Hi, I am Prathip Bavishi and I am going to talk about new technology, DNA, which is used for data storage. I am going to brief you about introduction and history of the DNA for data storage, the reason why we need this technology. Then I will go through the experiment done by Dr. Goldman. Then I will also explain you about its function, advantages, future challenges, future aspect of it, and finally I will come through the conclusion and the reference page. So what's the DNA for data storage? It refers to any scheme to store digital data in the base sequence of DNA. DNA is a robust way to store information because we can extract it from bones of woolly mammoths which date back tens of thousands of years and make sense of it. As we all know, DNA holds the genetic code of each species and spells out the exact instructions required to create a particular organism. The information in DNA is stored as a code made up of four chemical bases: adenine, guanine, cytosine, and thymine. It's also incredibly small, dense, and does not need any power for storage, so shipping and keeping it is easy. The history behind this technology goes this way. Dr. Nick Goldman and his colleague Ivan Berni from the European Bioinformatics Institute cleaned up the solution over a few years one evening. Their work was published on January 23, 2013 in the journal Nature. Nearly 60 years after Watson and Crick published the landmark paper that detailed the double helical structure of DNA in the same journal on April 25, 1953. Previous attempts at encoding data onto DNA failed because those methods tried to translate a computer's binary data directly onto the DNA. The binary repetition caused errors in retrieval. These were two challenges. First, using previous methods, it was only possible to manufacture DNA in short sprints. Secondly, both writing and reading DNA were prone to errors, particularly when the same DNA letter is repeated. Dr. Nick Goldman and his team was able to reproduce all the information from DNA files with 99.99% to 100% accuracy. The question is, why we need this technology? Companies, governments, and universities face an enormous challenge storing the ever-growing flood of digitized information, the videos, books, movies, and songs sent over the internet. Like the European Bioinformatics Institute generates a huge amount of data, and data storage is a real concern. But experiments conducted by Dr. Goldman changed all. The sequences of the individual strands of DNA overlapped in such a way that each region of data was repeated 4 times to avoid errors. Two of these four strands were constructed backwards, also with the goal of eliminating errors. Dr. Goldman and his team were able to encode and decode 5 computer files, including an MP3 recording of part of Martin Luther King's "I Have a Dream" speech, a PDF version of the 1953 paper by scientist James Watson describing the structure of DNA, and Shakespearean sonnet. The clips of Dr. King's speech and Shakespearean sonnets were played back. It sounded just like the original version. Then how does it work? It, it goes like this. Any data which is in computer is made of a long string of ones and zeros. There is a software program that converts those ones and zeros into ternary information which uses 0, 1 and 2. You can look in this figure, the whole function has been explained here. First there is zeros and ones produced by the computer, then it is converted into the ternary, and they also convert it to ACGT terms. I will describe it in detail in next slide. Then they store in the DNA, then it goes to the converter which converts ACGT format to the binary, and it goes back to the computer. The ternary information encoded into the letters A, C, G, and T as shown in the figure. There is a table given how they convert ternary into the ACGT format. They literally tried not to repeat anything, like if A is coming, then there will be C or G or T, but no other A will repeat after that. Then we will finally have the information in the A, C, G and T chemical bases, and that also makes up the DNA. You can see here A, C, G, T format, how they form a DNA. This is a figure for it. The single long DNA based string is broken into about 150,000 fragments, each 120 letters long. Each fragment is contain about 100 letters encoding the data. The remaining 20 letters serve as an index instruction for later restoring the fragments in the right order. The DNA lay like a speck of dust at the bottom of the vial, almost impossible to see. There a DNA sequencing machine fires lasers at the fragments and reads their genetic code, yielding a computer file in the form of A, C, G and T. The computer program reassembled the fragments in the right order and then converted them back into zeros and ones. When run on a laptop, those ones and zeros were interpreted as a normal data, like audio or sonnet. There are many advantages of this technology. The researchers think their system might be able to store the roughly 3 zettabytes, 3 zettabytes, which is 1 billion trillion bytes of digital data through to presently exist in the world. It is very expensive and inefficient to store data on hard disk. They need a constant supply of electricity, and over time the machinery degrades and ultimately fails. While the best achieving material currently available that doesn't rely on a power supply degrades in only a decade or so. That is of course DNA for data storage. It also has the capability for longevity. As long as the DNA is held in cold, dry and dark conditions, as is shown by the study of woolly mammoth DNA from up to 60,000 years ago, and for resistance to obsolescence, as DNA is a universal and fundamental data storage mechanism in biology. Archives could last for thousands of years when stored in DNA instead of magnetic tapes and hard disks. The technology is very promising, especially as a means to cut down on the cost of storing large amounts of data. It should definitely end up as a cost saver when actively long-term storage is needed. But it will also going to face some future challenges, because It needs to become a cheap and reliable commercial process. The researchers say that their next step is to work on perfecting the coding process and exploring the steps needed to commercialize the technology. And it is a slow process as the DNA needs to be sequenced in order to retrieve the data, and so the method is intended for use with a low access rate such as long-term archival of large amounts of scientific data. The other disadvantage is cost. Dr. Goldman estimates that at commercial rates, the method costs around $12,400 per megabyte stored. Thus, summing up all the possibilities in 10 years, it's probably going to be about 100 times cheaper. At that time, it probably will become economically viable. After going through this future technology, I would like to conclude three things. First, archives could last for thousands of years when stored in DNA instead of magnetic tape and hard drives. It can be very reliable source to store the digital data, and it does not need power supply. These are the websites from which I got this information: economist.com, earthsky.org, planetcell.com. Thank you for watching this video.